I'm going to today talk about a specific case that we've recently published of a complex Klebsiella pneumonia infection where we showed that real-time genomics, particularly nanopore sequencing, could help uncover hidden antibiotic resistance. So I don't have to tell anyone about the benefits of nanopore sequencing, especially in the clinical microbiology context. However, nanopore sequencing, with its ability to produce long sequencing reads, its ability for real-time data analysis, and its portability, has the potential to speed up pathogen identification, ensure plasmid typing, antimicrobial resistance profiling, and also strain-level detection, as we've heard yesterday morning in the talk. And this is especially relevant given the increase of infections with carbapenem-resistant Enterobacterales. So carbapenem is one of the last resort antibiotics, and there has been a rise in carbapenem-resistant Enterobacterales infections, which presents a significant challenge in the clinical setting because resistance tends to prevent or delay the administration of appropriate antimicrobial therapy, and in turn, the rapid administration of appropriate antimicrobial therapy, however, is the main prognostic factor of clinical outcome. Current established culture-based diagnostic approaches still rely on the bacterial growth and face several bottlenecks, such as turnover time, culture bias, and may miss key resistance factors. So I've shown here our cultural diagnostics approach in the University Hospital of Munich, and it might be similar in other hospitals as well. And as you can see, it spans several days, including incubation, subculturing, species identification using mass spectrometry, and antibiotic susceptibility testing by exposing the bacteria to certain concentrations of antibiotics and assessing the bacterial growth, again, requiring time-consuming bacterial growth. In contrast, nanopore sequencing has the potential to overcome some of these limitations by offering real-time genomic information on antimicrobial resistance. However, this still needs to, the accuracy of these predictions still needs to be compared to the current established methods, which is why we're currently in the University Hospital of Munich creating a comprehensive catalog of all carbapenem-resistant Enterobacterales routinely collected in 2023 to compare the performance of nanopore sequencing with regards to turnaround time and also information on resistance mechanisms to the current established culture-based approaches. And so far, we've seen a high consistency with clinically established approaches where carbapenemases as resistance mechanisms had been identified. In addition to the culture-based approach, we're also able to get more insight in the plasmid epidemiology of our hospital using nanopore sequencing. However, where it becomes even more interesting is inconsistent cases, such as cases where conventional diagnostic approaches could not identify any resistance mechanism, which is what I'm going to delve into, and which is what I've announced, it being the complex Klebsiella pneumonia infection case. So in this case, an immunocompromised patient presented with a fever in a clinic and was initially treated with the last resort antibiotic meropenem, which is part of the carbapenem subclass. The first sample of this patient grew a carbapenem-resistant Klebsiella pneumonia, with KPC, or Klebsiella pneumonia carbapenemase, being identified as resistance-conferring mechanism by means of lateral flow tests. So with this new information, the treatment was changed from meropenem to the even more last resort antibiotic ceftazidime avibactam, which was deemed to be susceptible in additional testing. After initial clinical improvement of the patient, however, the clinical condition deteriorated, and now consecutive blood cultures grew again a Klebsiella pneumonia. However, this Klebsiella pneumonia now showed a very rare and unusual resistance profile with a switched around resistance profile with restored in vitro carbapenem susceptibility and ceftazidime avibactam resistance. And I can't emphasize enough how rare this kind of resistance profile is. So by the time we had this information, the clinicians added meropenem to the antimicrobial therapy regimen. However, the patient unfortunately passed away shortly after, highlighting how crucial timely and targeted infection diagnostics can be. So to now simulate the advantages of nanopore sequencing for these complex resistance and infection cases, we set out to sequence both isolates, which I will now call pre-treatment and post-treatment isolate, referring to ceftazidime ivactam exposure. We sequenced them on R9 flow cells and MK1Bs using the rapid barcoding kit. And using our real-time genomic technology, we were able to accurately identify Klebsiella pneumoniae in the pre- and post-treatment sample as a putative pathogen based on the epitome antimicrobial resistance workflow. When looking at the resistance for the pre-treatment isolates, we were able to accurately predict a carbapenem resistance profile with pinpointing KPC of subgroup 2 as the resistance conferring mechanism, and this is one of the most common resistance conferring mechanisms for KPCs. Now, crucially, for the post-treatment isolate, while it was resistant to ceftazidime ivactam, the clinically established approach did not detect any resistance conferring mechanism shown here, with the lateral flow test coming back negative. So we had no information on the resistance conferring mechanism here. However, with our real-time genomics approach for the post-treatment isolates, which had this very unusual resistance profile, we were able to accurately predict this unusual resistance profile and, furthermore, pinpoint the resistance conferring mechanism in this case, which was KPC of subtype 14. Where we found quite a few alignments with high accuracy. And KPC14 is a relatively new KPC subtype, which was recently described to be associated with restored in vitro carbapenem susceptibility and ceftazidime ivactam resistance. So accurately predicting this phenotype, but going beyond clinical diagnostics, also looking at the resistance conferring mechanism. Now, crucially, when looking at the pre-treatment isolate, actually, we found the same one alignment of the same KPC14 gene already in the pre-treatment isolate. Now, to simulate the adaptive nature of nanopore sequencing in the clinic, we sequenced this pre-treatment isolates even more deeply to see if this KPC14 copy is confirmed. And indeed, with 8 hours of additional sequencing, we've detected 4 additional copies of KPC14 on the pre-treatment isolate already. Now, we argued that potentially we were able, with real-time genomics, to show evidence of low abundance pre-existing ceftazidime ivactam potential prior to ceftazidime ivactam exposure, which might have guided a different antibiotic regimen to start with. Now, we've also been able to create fully circular de novo assemblies of the chromosomes and 3 plasmids, and core genome multi-locus sequence typing showed that both Klebsiella pneumoniae and the pre- and post-treatment isolates belong to the same strain, which is a sequence type 147 strain, which is a high-risk and emerging strain. So this might indicate that we are actually dealing with a single-strain infection. When zooming into and functionally annotate the plasmids, we were able to detect that both KPCs were located on the ink and plasmid. And the ink and plasmid of the pre- and post-treatment isolate showed a lot of genetic similarity. We've also looked at the increase of normalized abundance of KPC14 before ceftazidime ivactam treatment and after, as it encodes ceftazidime ivactam resistance, and we're able to detect an increase in normalized abundance from 0.56 to 26.6% after ceftazidime ivactam exposure. So to summarize, the added value of nanopore sequencing in the clinical context now is for the pre-treatment isolate, we were able to accurately predict and pinpoint the resistance conferring mechanism, but going beyond that, we were able to also identify a low abundance KPC14 gene, which might indicate the potential of KPC14 selection under antibiotic selection pressure. For the post-treatment isolate, resistance and resistance mechanism were accurately predicted, and we had a shorter turnover round compared to established diagnostic approaches. So this highlights, to summarize, the potential impact of real-time genomics on clinical management, such as this specific clinical case, by offering faster and now also, in this case, more accurate resistance predictions in the clinic. Thanks a lot for your attention. And especially thanks a lot to my supervisor, Lara Urban. Thank you.